Hello learners, welcome to the online course on functional foods and nutraceuticals. I am Dr. Rekha Sharma, Associate Professor UGC Human Resource Development Center, Rashtrapati Tukaroji Maharaj, Nagpur University, Nagpur. In this module, we shall study protease inhibitors. Plants are good sources of protease inhibitors which protect them against diseases, insects, pests and herbivores. In the past, proteinaceous protease inhibitors were considered primarily as protein degrading enzymes. Nevertheless, this view has significantly changed and protease inhibitors are now treated as very important signaling molecules in many biological activities such as inflammation, apoptosis, blood clotting and hormone processing. In recent years, protease inhibitors have been examined extensively as therapeutic agents, primarily to deal with various human cancers. Interestingly, many plant-based protease inhibitors are also found to be effective against cardiovascular diseases, osteoporosis, inflammatory diseases, and neurological disorders. Among all types of protease inhibitors, Bowman-Burke inhibitors have been studied extensively in the treatment of many diseases, especially in the field of cancer prevention. Protease inhibitors have been found in a great variety of plants, including most legumes and cereals and certain fruits like apples, bananas, pineapples and raisins, and vegetables such as cabbage, cucumbers, potatoes, spinach and tomatoes. The quantity of protease inhibitors depends on variety and physiological status of the plant and on levels of insect infestations or damage. Let us learn more about protease inhibitors in this module. Now, what is protease? Proteins are made up of individual amino acids that are linked by a covalent bond called a peptide bond during the process of protein translation. Proteins are constantly being translated and degraded in the cell to ensure adequate concentrations of functional proteins to complete various cellular functions. The process of protein degradation, referred to as proteolysis, breaks protein down into individual amino acids. Proteolysis is accomplished by proteases, a type of enzyme whose mechanism depends on the particular class of protease. Now, let us see what are proteolytic enzymes. Proteolytic enzymes are also called as protease, proteinase or peptidase. Any of a group of enzymes that break the long chain like molecules of proteins into shorter fragments, that is peptides, and eventually into their components amino acids. Proteolytic enzymes are present in bacteria, archaea, certain types of algae, some viruses and plants. They are most abundant, however, in animals. There are different types of proteolytic enzymes which are classified according to sites at which they catalyze the cleavage of proteins. The two major groups are the exopeptidases, which target the terminal ends of proteins, and the endopeptidases, which target sites within proteins. Endopeptidases employ various catalytic mechanisms. Within this group are the aspartic endopeptidases, cysteine endopeptidases, glutamic endopeptidases, metalloendopeptidases, serine endopeptidases and threonine endopeptidases. The term oligopeptidase is reserved for those enzymes that act specifically on peptides. Proteolytic enzymes are ubiquitous in all organisms and constitute 2 to 4% of the encoded gene products. They are critical for diverse biological processes such as digestion, blood clotting, host defense, pathogenic infection, viral replication, wound healing and disease progression, to name a few. Because proteases trigger an irreversible event, the cleavage of a protein, their activity must be tightly controlled. Dysregulated proteolytic activity causes a disruption in the homeostatic balance of a biological system and can result in any number of poor biological outcomes. As a result, nature has developed a number of strategies to control proteolysis, including spatial and temporal regulation, zymogen activation and protease degradation, and through the inhibition of proteases by macromolecular inhibitors. Recent studies have shown that proteases are also involved in progression and tumor growth both at primary and metastatic sites. A positive correlation between the aggressiveness of tumor and the secretion of various proteases has been found. Proteases in normal cells are very important in carrying out important biological processes, but transformed tumor cells are the cause of heavy destruction. The secretion of some specific proteases in tumor cells also make prognosis very difficult. However, proteases are not exclusively expressed by cancer cells. Now let us see what are protease inhibitors. Protease inhibitors are molecules that block the activity of proteases and typically function on classes of proteases with similar mechanisms of action. Protease inhibitors can either be in the form of proteins, peptides or small molecules. Naturally occurring protease inhibitors are usually proteins or peptides. In medicine, protease inhibitor is often used interchangeably with alpha-1 antitrypsin or A1AT, is indeed the most often involved in disease, namely in alpha-1 antitrypsin deficiency. Now let us study the classification of protease inhibitors. Protease inhibitors may be classified either by the type of protease they inhibit or their mechanism of action. In 2004, Rawlings and colleagues introduced a classification of protease inhibitors based on the similarities detectable at the level of amino acid sequence. This classification initially identified 48 families of inhibitors that could be grouped into 26 related superfamilies. Now, according to MEROPS database, there are now 81 families of the inhibitors. Now, the classification of protease according to the classes of proteases. They are aspartic protease inhibitors, cysteine protease inhibitors, metalloprotease inhibitors, serine protease inhibitors, threonine protease inhibitors, trypsin inhibitors like Conid's STI protease inhibitors. By mechanism, they have been classified as suicide inhibitors, transition state inhibitor, protein protease inhibitor, that is serpins, and chelating agents. Now let us summarize. We have studied proteases, protease inhibitors and their classification.